I always got your name wrong. Please share your screen and we can go on. And yeah, I just have a call out maybe to the IHP staff. If you could make Mikhail Tikhonov co-host, please. All right. So yeah, our next speaker is Florence Debarre. So go ahead.
Okay, good afternoon or good morning, everyone. So today I'm going to present the results of models on the spread of gene drive and ways of stopping them over space. So there are some animal species that we try to conserve and save, and there are also other animal species that we humans would be happier if not without, at least in lower numbers, because they interfere with our activities or our health, or also because we think that they are dangerous for biodiversity in general in the case of invasive populations. And to fight against pests, there are different ways that are usually classified as follows. So there is mechanical or physical pest control, like the use of bed nets against mosquitoes. There is chemical control, like the use of insecticides or more generally biocides, and biological control, the use of natural enemies. And within biological control, there is a subcategory that's called genetic control, and that's defined as the dissemination by mating or inheritance of factors that reduce pest damage. There are different types of genetic control, and you've probably heard about already the sterile insect techniques because it's been used since the mid-20th century in the field already. And it relies on the release of sterilized males of the species that you want to reduce the numbers of. And then wild-type females try to mate with them and don't produce offspring. So in the traditional technique, the males are sterilized by irradiation. Now there are more recent techniques that use genetic modifications, and they've been in the press recently again. I'm mentioning it just to warn you that this is not what I'm talking about today. And today I'm talking about another method of genetic control of populations that is called gene drive. And that is the use of selfish genetic elements, that is elements that are transmitted to the offspring more often than you would expect under regular Mendelian segregation, which is illustrated here with this genealogy. And the reason why I'm talking about it is that even though it's a quite old idea, it has recently been made possible. And it has become possible thanks to the use of the CRISPR-Cas machinery that has been applied to genomic addition, but can also be used for gene drive. And the way it works is the following. So you insert a gene drive cassette, and then when you have a heterozygous cell with the gene drive cassette and the wild type sequence, then the Cas9 endonuclease associates with a guide RNA, locates a target sequence on the wild type chromosome, which induces double strand DNA cutting. And then you have repair mechanisms that use the other chromosome as a template and leads to the duplication of the gene drive cassette. And I'm going to refer to this process as gene conversion. You can envision different types of drives. So gene drive to modify a population, so to spread a new trait in a population, for instance, in mosquitoes, making them resistant to Plasmodium, the agent of malaria. And there are also eradication drives whose aim is to, suppression, or eradication drives, whose aims are to reduce the numbers of the target population or even eradicate it. And also something that matters is the timing of gene conversion. So when the duplication takes place. So it can be programmed to take place very early in development, so that individuals that were initially born, well, created heterozygote immediately become homozygote and spend their lives as such. Or gene conversion can take place very late during gamete formation. And then the individuals carrying them remain mostly heterozygote. And in this talk, I'm going to focus on the first case. So if we want to model a gene drive of this type, so you have two types of alleles, the wild type allele and the drive allele. Then you have three genotypes and associated fitnesses, denoting by S the cost of the drive, and C conversion efficiency, that is the percentage of the time that you go from this heterozygous state to the homozygous state. And then you can write the usual recurrence equations if you assume that you have a well-mixed population with synchronous generations. And the point that makes this model different from a model of classical Mendelian transmission is what happens for heterozygotes. And if you assume that gene conversion takes place very early in development, then when it does take place, the individuals are essentially homozygotes, so have the fitness of homozygotes, and all of their gametes carry the drive allele. While when gene conversion doesn't take place, the individuals are heterozygotes, have the fitness of heterozygotes, and only half of their gametes carry the drive allele. Then if you want to also follow population size, you can have an equation for that. And essentially, there are three types of equilibria in this kind of model. So either the drive allele is lost, or it's fixed, or there is an intermediate frequency, which can be stable or unstable. And then you have different combinations of parameters leading to the different outcomes, and the ones we're interested in are the ones where eventually the drive can be fixed and do its job in the population. And so what's going to be very important in what follows is the distinction between these two regions. So when the drive can be fixed and it increases from any initial frequency, and a bistability regime where the drive cannot increase if it's introduced into low frequency. And also I want to highlight the fact that the transition between these two regions is at 0.5 when the conversion efficiency is 1. So just that's an illustration of what I've showed before, the time dynamics of, in red, the frequency of the drive, and in black, the size of the population. So when you're in the parameter regime of drive fixation, that's also called threshold independent drive, then the drive can spread in the population, go to fixation. And in the meantime, the size of the population decreases to the point under these parameters that the population goes extinct. So you can indeed have population eradication in theory. When you are in the bistability regime, it's a bistability. So you need to introduce the drive in a high enough frequency for it to fix. Otherwise, it gets lost. So what I've presented so far is for a well-mixed population, a single well-mixed population. And now we can ask the question of the spatial spread of a gene drive. So it's been covered by previous authors. And what they did is they used the reaction diffusion framework. They assumed perfect conversion. So the C parameter is 1. And they, their reaction term is basically the same equation as I presented before when C equals 1. And then it's put in a reaction diffusion framework. And what they've shown is that if, so the cost of carrying the drive is lower than 0.5, which with what I presented before corresponds to the drive fixation regime, then you have a pooled wave and the drive can spread over space. If S is greater than 0.5, you have a pushed wave regime and you need a big enough quantity of drive initially, so over space, for the wave to actually spread. And then if the cost is really too high, the wave reverses direction and the drive doesn't spread and gets lost, no matter the introduction frequency. And also what's important in the difference is that you can stop a pushed wave with physical barriers while you can't with the pooled wave. So that's one way potentially of stopping a gene drive. Another one is using a brake system. So some people have proposed molecular brakes that could be introduced in a population to stop a gene drive if you had to. And then the way it works is that it uses a guide RNA against Cas9 and then targets Cas9 and then cuts, and the brake gets reproduced, well, duplicated and replaces the drive. You can think of different types of brakes that do or do not restore fitness. But the most important point here is that you end up with rock, paper, scissors dynamics, so non-transitive interactions, because drive wins against wild type, brake wins against drive, and brake loses against wild type if you assume that there is some cost of carrying it. And so we asked, can an artificial gene drive be stopped by a brake construct? So with collaborators, we asked this question in two settings, well-mixed population and spatial spread. And today I'm only talking about the spatial spread aspect of it. And that was done with Vincent Calvez and Léo Girardin, who are two mathematicians. And so this is our model. It's exactly the same idea as what I took the time to present before. So I'm going to be much faster here. The only thing is that we have a third equation for the frequency of the brake in the population. And as previously, to make things simpler, we assume that gene conversion is perfect. And so with this model, what happens? So I'm going to show that with small videos. So here, so in this first case, so that's the, oops, sorry. I hope you're not too seasick. Okay, I cannot move the, that's interesting. Okay, I'll try to do that anyway. So in red, that's the frequency of the drive over space. And in black, you're going to see the brake over space. So initially the drive spreads. Then the brake is introduced, catches up, and you have an everlasting chase with odd dynamics in the back. So here we were in the fixation regime. And in this case, the brake cannot stop the drive and you have a never-ending chase over space. If you are in the bistability regime, so again, in red, it's the drive. In black, it's going to be the brake. What's happening? Okay, so the drive spreads over space, then the brake is introduced, and it catches up and it's over. So what's over is that I'm not showing the third frequency, which is the frequency of the wild type. And then you only have wild types in the end. So basically here, what happens is that you have this P stability. So you have this critical threshold frequency of the drive in the population, below which the drive gets lost. And the introduction of the brake can make the drive frequency go below this threshold and then be lost. And so we can recover a wild type population in this case. So to answer our question, can an artificial gene drive be stopped by a brake construct? So in this ideal deterministic model, you only can under the P stability regime, and that's the same conclusion in spatial and non-spatial models. But actually then, if you consider a bit more realistic settings, which we investigated with a well-mixed population, then a lot of factors matter, especially because you have these high amplitude oscillations that take place and reach unrealistic frequencies, unrealistically low. And so as soon as you have a finite population, you can have loss of an allele. So now to conclude this talk, a question that we may ask is, will a brake actually be necessary? Because gene drives may not work as advertised. Resistance can evolve, resistance to the drive in the target population. And you can also have other effects of evolution, like the evolution of mating systems, to reduce the proportion of heterozygotes in the population and thereby the effect of the drive. We can also ask whether gene drives should be used in the first place. So here I'm going to gloss over this very quickly in the interest of time, but this should be an entire talk or a day of workshop. So because there are risks associated to the use of gene drive, risks of off-target effects, off-target off-target populations, and even maybe off-target species. There's a host of regulatory issues because gene drives, animals or plants actually with gene drives are living genetically modified organisms and as such are regulated. But they are differently regulated in different parts of the world. And there is also a whole host of ethical issues, including the fact that so far genetic modifications were on domesticated species, and here we're talking about genetic modifications affecting natural populations. So with this, I've reached the end of my time, and I thank you for your attention.
Thank you very much for this very interesting talk. Questions?